Latest research on hepatitis B cure. As per World Health Organization, there are around 296 million people live with a chronic HBV infection worldwide that is often asymptomatic until liver disease has advanced so far that it is largely untreatable. HBV has long evaded effective therapeutics. As a result, nearly 1 million people die from HBV every year. As per the report of Rockefeller University, developed an approach for studying HBV in the lab that sidesteps the virus's typical replication process, allowing for a much sharper view of its behaviors and characteristics during a crucial part of its life cycle. Dr. Charles M. Rice, Nobel Prize winner in physiology or medicine for pioneering novel methods to grow and study HCV. He added HCV exposed that virus's weaknesses. The hope is that this novel approach could do the same for HBV. Bill Schneider, a research associate in Raya's Laboratory of Virology and Infectious Disease, he shared it's important to study the whole virus life cycle, because every step is important for how the virus spreads and infects new cells. The Raya's Laboratory of Virology and Infectious Disease, research on HBV and HCV are quite different. Both viruses are totally different from each other and have different genetic compositions. However, HBV is a DNA virus, whereas HCV is a RNA virus. HBV is only about 1/3 the size of HCV, means very hepatitis B virus is very small than hepatitis C virus with a unique genetic architecture that makes it very challenging to study so far. Both viruses do share some characteristics. Both are easily transmittable and hard to shake once an infection has taken root. In case of current HBV therapies, can cause intolerable side effects or have limited impact, leading to lifelong drug therapy. Now some advanced therapeutic vaccines are available which are very effective and that can block new HBV infections. Why vaccine development progress research is going slow? The vaccine development progress is slow for HBV due to some reasons including: first, HBV is especially difficult to culture in the lab. It's not clear why, because it's an extremely effective virus in nature. Schneider says, with the origins dating to 400 million years ago, HBV and its relatives are capable of infecting a variety of animals. Second, HBV in the lab is plagued by background noise from HBV DNA and plasmids. The result is a lot of genetic fuzziness that makes it hard to clearly see the virus's properties. Third, Yingpu Yu, a research associate of the team, he added, there is so much noise from the plasmids that is difficult to distinguish something made from the replicating virus as opposed to the plasmid. He also said, it's like throwing a lit candle into a bonfire and then trying to study the candle flame. Fourth, the whole scientists team are working hard and tried workarounds to tackle this problem. But Rice hit on a winning idea: trigger the virus's life cycle later in the process using RNA, which might allow them to avoid all the DNA noise. How chronic hepatitis B works after infected in the liver. HBV hijacks a cell's molecular machinery in order to reproduce, but its process is a bit unusual. Once inside the nucleus, it uses that machinery to first transcribe its DNA to RNA, and then converts into a new viral genome called cccDNA. These cccDNA genomes that are extremely difficult to get rid of. cccDNA is a special DNA structure that arises during the propagation of some viruses in the cell nucleus and may remain permanently there. cccDNA is a double-stranded DNA that originates in a linear form that is ligated by means of DNA ligase to a covalently closed ring. In most cases, transcription of viral DNA can occur from the circular form only. This virus is also known as episomal DNA. If they could start HBV's life cycle before it produces cccDNA, that might give them an unobstructed view of the process at the heart of HBV's ability to become a chronic, damaging tenant of the liver. The great thing is, HBV's tiny size, it has few RNA transcripts involved in the replication process. One turned out to be key, called as pre-genomic RNA. In the previous research, the PgRNA was able to instigate replication in a relative of HBV that infects ducks. They wondered if they could use it to spark the replication cycle in human HBV too. However, the team has seen it worked. Through cultured cells and then in mouse models, they were able to kickstart production of the virus's cccDNA using its RNA, thereby silencing the background noise. The new platform has great potential to play a role in the development of new therapies or a cure for HBV. Finally, Raya's method worked with HCV. But still, much more research is going on and needed to do for HBV. Schneider says, the life cycle and prevent of this virus from replicating and spreading to new cells could be a potential target for new drugs. Yet it's not clear what is the right combination of therapies will look like. All we know at the moment is that the ones that we have aren't doing the job. If you like this video, please like, share our videos as well as press the bell icon to get the new video updates instantly. If you have any queries or know about advanced research on hepatitis B treatment, please share in the comment box. Thanks for watching Great Wisdom of Health. Note: the information presented in this video is for general information only and should not be taken as professional medical advice.